I started Carolina BioOncology Institute in 2005 after being in private practice for several years. I strategically chose the location because of access to our local hospital across the street, Novant Medical Center, and access to expand our business as it grows, along with the Charlotte area, its infrastructure, and international airport. Immunotherapy is an incredibly promising field. It's now at the cutting edge. I've developed over 80 phase 1 clinical trials over the past 14 years. While during my medical school at Georgetown University, I spent my senior year with Dr. Steve Rosenberg, considered one of the grandfathers of immunotherapy. That's where I got the bug early in my career. After that, I performed my residency at University of Texas, Houston Health Science Center, did research at MD Anderson, focusing on tumor immunology, and then did my fellowship at University of North Carolina at Chapel Hill, further focusing on tumor immunology and melanoma. That's a good question. I'll tell my mom you thought I was that young. I'll compliment your wife too. She looks young. We say phase one research is first in human. So all the drugs that we would be offering you on trials, those drugs are so new, they don't even have a name. They're still just a number. I became triple board certified in medical oncology, internal medicine, and also pediatrics. And today we run a cancer clinic focused on developing phase 1 drugs for access to cancer patients in our community setting. Over the past 14 years, we've developed many strategic relationships with our biotech and biopharma partners. And we continue to be nimble and focused as an independent cancer center. In fact, we're the only independent phase 1 community-based cancer clinic in the country.
As leaders in early phase clinical trials in immunotherapy, our clinical research program is specifically designed to meet the needs and complexities of phase 1 clinical trial design and drug development. Our hand-picked staff of experienced medical research professionals, fast accrual, coordinator and investigator responsiveness, and accelerated startup has proven to be highly valuable. Our mission is to provide the highest level of comprehensive and quality medical care possible. To accomplish our mission, we work as a team with our patients' health and safety as our common goal. We are constantly striving to improve, rededicating ourselves daily to a set of values we committed to when we joined this cancer fighting team.
Hi, my name is Tori Prevet, and I am the leukapheresis supervisor here at Carolina BioOncology Institute. During leukapheresis, we extract white blood cells for donation while returning the remaining blood to the patient. We are performing leukapheresis to collect lymphocytes, to research how to grow those lymphocytes and activate them in order to recognize and destroy cancer cells. Our long-term goal is to help our own patients fight their own cancer with their own lymphocytes.
At the same time in 2005, I also started a different organization called BioCytics. So BioCytics first means applications of living cells. Specifically, we're developing T cells. T cells from patients' own blood, autologous, meaning they can be grown up, trained to fight cancer patients' tumors.
I'm Amy Austin. I'm the Human Applications Lab Manager and Clinical Lab Manager for the clinic. I lead a highly skilled group of individuals, including cell processing specialists, flow cytometry specialists, biomedical engineers, laboratory technicians, and laboratory research coordinators. We operate a wide array of different instruments for both small-scale and large-scale projects. This includes devices that perform apheresis, closed system cell selection and expansion, cell separation and washing, incubation, cell analysis, closed system flow sorting, and flow cytometry. We also partner with device makers to help them learn more about their devices and bring the full potential of their inventions to market by performing beta testing and studies. We recently expanded to include space for a large ISO 7 clean room in order to provide GMP cell therapy manufacturing, along with space for future USP 797 and USP 800 pharmacies.
My name is Midan Ai. I'm a tumor immunologist working in the Human Application Lab of Carolina BioOncology Institute. My work focuses on developing new therapy for cancer patients. The expression of PD-1 rate will happen on the turn on the T cells use the anti-PD-1 and anti-PD-1 signal. Our work mainly focuses on the preclinical study to develop a new therapy for cancer patients.
Right, so you want to pick kind of the best one from them. I work together with the Human Applications Lab and the Tumor Immunology Department to develop our own T-cell cellular immunotherapies. The goal of my project is called the ex vivo lymph node. The goal of this project is essentially to reprogram and reinvigorate the patient's immune system. We want to take out the T-cells from the patient, use autologous monocyte-derived dendritic cells to teach those T-cells what to target, to teach them to become tumor-specific. We'll then reinfuse those T-cells back into the patient, and in this way we're using autologous patient cells. We're teaching them how to be tumor-specific, reinfusing them, and then allowing those now educated T-cells to attack that patient's tumor. In this way, we're not using any xenogenic cells, any foreign materials. We're simply reinvigorating the patient's immune system and teaching it how to overcome the cancer that has shut it down.
We have just completed our ISO 7 Class 10,000 clean room, where crucial systems are backed up by dual supply, like the gas system next to me. And we also have a generator on site that provides power in case we lose power for critical systems. Overall, that is a Reese monitoring system that is looking at 50 points constantly and allows us to take a look at trends so we can spot problems before they occur, but also will call someone if someone is needed to attend to a problem. We have a dedicated space for our USP 797 and our USP 800 pharmacies, and we're also working on the next expansion, which will have additional clean room and GLP space. We're a small company, but we're also fast and we're agile, so we can meet the demands of a changing business in immunotherapy.
People ask, why did you start this dual model clinic and research lab? Really for two reasons. One is pure frustration with the status quo because cancer research moves too slowly. Administrative delays, bureaucracy, high cost, and I saw that firsthand. So I wanted to address that and fix it. I wanted to take personal responsibility to bringing cutting-edge research to the bedside to patients in a more efficient, rapid way that is deployable and clonable. So that was the reason we started it, was frustration. Second reason I started it was inspiration. Cancer patients inspire us. They're fighting for their life. What an honor. They're volunteers on clinical trials. The least we can do is get treatments to them fast, without bureaucracy or administrative delays, and bring costs down, and to do that at the point of care. And that is inspiration for me, at a minimum, to die trying to try to save a cancer patient's life.